Muscles are composed of contractile tissue that is capable of changing its length by contracting and relaxing. Most animals rely on muscle tissue to produce movement, to swim, crawl, walk, run, jump, or fly. Clearly, there are strong advantages to being able to move into new environments, to flee from danger, to seek food, and to find new mates. Only few animals are known motile, also called sessile, and most of these live in water where currents can bring a supply of food to them. Humans and other vertebrates have three distinct types of muscle tissue: smooth, cardiac, and skeletal. Most of the focus in this section will be on skeletal muscle or striated voluntary muscle, which is important in maintaining posture, providing support, and allowing for movement. The processes responsible for skeletal muscle contraction also release heat, which is distributed throughout the body and helping maintaining a constant body temperature.
In this topic, we have microscopic anatomy and sociology. For your information, 1650 skeletal muscle and their associated tissue has made up 40% of the weight of our body. Muscle tissue is 50% more dense than fat tissue. However, at the rest, muscle tissue consumes about 3 times energy than adipose tissue. Next, antagonistic muscle. Skeletal muscle must work in antagonistic pair because when muscle contract, they shorten, so they can only pull and cannot push. For example, the bicep and tricep are antagonistic pair because they act opposite to one another. The bicep flex elbow joint and the tricep extend the elbow joint. Okay, when our hand flex, the bicep will contract and the tricep will relax. While our hand extend, the tricep will contract and bicep will relax. So they act opposite to one another. Tendon is skeletal muscle that attach to the skeleton by bands of the fibrous connective tissue. Tetany is one of the illnesses that related to the muscular contraction. The involuntary muscular contraction and alteration in our brain cell are the symptom of this illness that caused by the low of calcium level in our blood. For example, when in hypocalcemia, the threshold needed for the activation of nerves will decrease and the neuromuscular activity will increase. Therefore, it will make neurons become unstable and fire spontaneous action potential that trigger the involuntary contraction of the muscle. Next, muscle tone. Muscle tone is the tension in our muscle at the rest. The sensory receptor called muscle spindle and Golgi tendon are responsible in maintaining our muscle tone. This is important for maintaining our posture. Okay, we can eat vitamin D and protein such as fish, chicken, and egg to maintain our muscle tone.
I will be presenting about microscopic anatomy and physiology. First and foremost, you must know that muscle is very important in an organism. It helps us move and do our daily activities. In understanding how the muscle works, you must first understand the anatomy of muscle. For instance, let's say that this is a bicep muscle. The whole muscle here is we call as belly muscle. Inside the belly, there are a lot of fascicle, which is bundle of muscle fiber. Inside the bundle of muscle fiber, there are a lot of individual muscle fibers. Inside the individual muscle fiber, there are hundreds or even thousands of tiny myofibrils. Now, we focus on the individual muscle fiber. As we can see in this diagram, there are many organelles that present here. Some of the crucial components are the sarcolemma, sarcoplasmic reticulum, and myofibril. Sarcolemma or plasma membrane form key system to make the muscle in contact with the sarcoplasmic reticulum. The sarcoplasmic reticulum is expanded portion of modified endoplasmic reticulum, which act as storage for calcium ion. Myofibril is the contractile portion of muscle fiber. Next, we go to the myofibril. Myofibril are cylindrical and run along the length of muscle fiber. Light microscope show that the myofibril is striated. It have dark band and light band. Sarcomere are the contractile unit within the muscle fiber. A unit of sarcomere are between the Z line. I band is light colored because only 18 filament attached to the Z line. A band is darker in color because actin and myosin filaments are overlapping each other, and H band only has myosin filament. Sliding filament model. Examining muscle fiber when they are contracted reveal that the sarcomere within the myofibril have shortened. When a sarcomere is shortened, the actin, which is thin filament, slide past the myosin, the thick filament, and approach one another. This caused the I band to shorten and the H zone to nearly or completely disappear. The movement of actin filaments in relation to myosin filament is called the sliding filament model of muscle contraction. During the sliding process, the sarcomere shorten even though the filament themselves remain the same length. When you play tug of war, your hand grabs the rope, pull, let go, attach further down the rope and pull again. The myosin head are like your hands, grasping, pulling, letting go, and then repeating the process. The participant in muscle contraction have function listed in the table here, which is actin filament. The function is slide past myosin causing contraction. Calcium ion needed for myosin to bind to actin. Myosin filament pull actin filament by means of cross bridges and are enzymatic and split ATP. ATP, the function is to supply energy for muscle contraction. Although the actin filament slide past the myosin filament, it is that the myosin filament that do the work. Myosin filament break down ATP and form cross bridges that attach to and pull actin filament toward the center of the sarcomere.
ATP provides the energy for muscle contraction. Although muscle cells contain myoglobin, a molecule that stores oxygen, cellular respiration does not immediately supply all the ATP that is needed, while muscle fibers rely on creatine phosphate, which is a storage form of high energy phosphate. Creatine phosphate also cannot directly participate in muscle contraction. Instead, it anaerobically regenerates ATP by the following reaction. When all of the creatine phosphate is depleted, mitochondria may then be producing enough ATP for muscle contraction to continue. If not, fermentation is a second way for muscles to supply ATP without consuming oxygen. Fermentation, which is apt to occur when strenuous exercise first begins, supplies ATP for only on a short time. We all have had the experience of needing to continue deep breathing following strenuous exercise. This continue intake oxygen, which is required to complete the metabolism of lactate and restore cells to their original energy state, offsets what is known as oxygen debt. In persons, as we know, who regularly exercise, such as athletes in training, the number of mitochondria increase and muscles rely on them rather than on fermentation to produce ATP. Less lactate is produced and there is less oxygen. For muscle innervation, muscles are stimulated to contract by motor nerve fiber. Nerve fiber have several branches, each of which ends at an axon terminal in close proximity to the sarcolemma of a muscle fiber. Axon terminals contain synaptic vesicles filled with the neurotransmitter acetylcholine (ACH). When nerve impulse traveling down a motor, neuron arrive at an axon terminal. The synaptic vesicles release the acetylcholine into the synaptic cleft. Acetylcholine quickly diffuses across the cleft and binds to receptors in the sarcolemma. Then the sarcolemma generates impulse that spreads over the sarcolemma and down T tubules to the sarcoplasmic reticulum. The release of calcium from the sarcoplasmic reticulum causes the filaments in sarcomeres to slide past one another. Sarcomer contraction results in myofibril contraction, which is in turn results in muscle fibers and finally muscle contraction. Once a neurotransmitter has been released into a neuromuscular junction and has initiated a response, it is removed from the junction. When the enzyme acetylcholinesterase breaks the acetylcholine, muscle contraction ceases. Threads of tropomyosin wind about an actin filament, and troponin occurs at intervals along the threads. Calcium ions, which is Ca2+, that have been released from the sarcoplasmic reticulum combine with troponin. After binding occurs, the tropomyosin threads shift their positions and myosin binding sites are exposed. Myosin heads function as ATPase enzymes splitting ATP into ADP and P. These reactions activates the heads so that they can bind to actin. The ADP and P remain on the myosin heads until the heads attach to actin, forming cross bridge. Now ADP and P are released and this causes the cross bridge to change their position. This is the power stroke that pulls the thin filaments toward the middle of the sarcomer. When more ATP molecules bind to myosin's head, the cross bridges are broken as the heads detach from actin. The cycle begins again. The actin filaments move nearer the center of the sarcomer each time the cycle is repeated. Contractions will continue until nerve impulses and calcium ions are returned to their storage sites. The membranes of the sarcoplasmic reticulum contain active transport proteins that pump calcium ions back into the calcium storage sites, and muscle relaxations will occur.
Muscle tissue can change its length by contracting and relaxing. Skeletal muscle attached to bone via tendons. Whole skeletal muscle can shorten only when they contract, therefore they work in antagonistic pairs. A muscle at rest exhibits tone, meaning some fibers are contracting. Tetany refers to maximum sustained muscle contractions. A whole skeletal muscle is composed of muscle fibers. Each muscle fibers is a cell that contains myofibrils, in addition to the usual cellular components. The plasma membrane, sarcolemma, forms a transverse T system. T tubules penetrate the cell and contact the sarcoplasmic reticulum, which stores calcium ions. Myofibrils are divided into sarcomeres, which display the arrangement of actin and myosin filaments. The sliding filament model of muscle contraction states that myosin filaments have cross bridges, which attach to and detach from actin filament, causing actin filament to slide and the sarcomere to shorten. The H zone disappears as actin filament approach one another. Myosin breaks down ATP, and this supplies the energy for muscle contraction. During oxygen depths, anaerobic creatine phosphate break down and fermentation can quickly generate ATP. Sustained exercise requires cellular respiration for the generation of ATP. Nerves innervate muscle. A nerve impulse travels down a motor neuron to a neuromuscular junction, causing the release of ACH, which binds to receptors on the sarcolemma. Impulses begin and move down T tubules that approach the sarcoplasmic reticulum, while calcium is stored. Thereafter, calcium ions are released and bind to troponin. The troponin-calcium complex causes tropomyosin threads winding around actin filament to shift their position, revealing myosin binding sites. Myosin filaments are composed of many myosin molecules with double globular heads. When myosin heads break down ATP, they are ready to attach to actin. The release of ADP and phosphate causes myosin heads to change their position. This is the power stroke that causes the actin filament to slide toward the center of sarcomere. When more ATP molecules bind to myosins, the heads detach from actin, and the cycle begins again.